Welcome. This recording is about the INSPiRED project, which is an acronym for Inclusive Diagnostics for Poverty-Related Parasitic Diseases in Nigeria and Gabon. I am Michelle Bengtson from the Department of Parasitology at the Leiden University Medical Center in the Netherlands. The INSPiRED project is funded by NWO Wotro Science for Global Development, and the consortium is a collaboration of European and African partners geared towards contributing to the achievement of the Sustainable Development Goals. The consortium has 5 partners: the Parasitology Department from the Leiden University Medical Center, 2 faculties from the Delft University of Technology, Industrial Design Engineering and Mechanical Engineering, the Faculty of Health Promotion and Education from the University of Ibadan in Nigeria, the College of Medicine from the University of Lagos in Nigeria, and SAMEL, the Medical Research and Training Center in Gabon. We have 3 PhDs and 2 postdocs from 3 African countries: Nigeria, Cameroon, and South Africa. The objectives of the INSPiRED project are to reduce the burden of malaria, schistosomiasis, and hookworm in Nigeria and Gabon. And we aim to do this by combining the expertise from the consortium partners in technology, design, disease knowledge, and end user requirements. If you would like more information on the consortium partners, please refer to the website. Let's take a closer look at the current diagnosis of these poverty-related parasitic infections. Conventional microscopy is currently recommended by the World Health Organization. It is a highly specific method that is also quantitative. It is relatively easy to use and is considered field deployable. However, this method does lack sensitivity, especially with respect to light intensity infections. It is time consuming, training is required, and there is variation in egg output, so you need multiple samples and multiple slides on multiple days. The use of this method is prone to underestimate true prevalence and misinform mass drug administration programs. Therefore, there is a clear need to develop, evaluate, and implement optical diagnostic devices that are easy to use, affordable, and effective against poverty-related parasitic infections that are of public health importance. And the INSPiRED project aims to do this through distinct work packages. Work package 1 deals with the hardware and software developments related to sample preparation as well as the diagnosis. Work package 2 focuses on the healthcare system and identifying relevant stakeholders through the use of co-creation tools. Work package 3 relates to laboratory and field testing and validation of the devices that are developed in work package 1. And the outcomes of the 3 work packages will be integrated into action towards implementation in work package 4. And again, we will be leveraging on the expertise from all of the consortium partners. I would now like to share with you our midterm progress in this project. With regards to the technology development, hardware and software, initially the project aimed to focus on malaria, but as the project has evolved, we also now move towards other blood parasites such as microfilaria. For the detection of these blood-based parasites, we use the ASSIST device. And for sample preparation, we have also developed a prototype of a blood smear kit. In terms of automated image processing, we are currently developing dedicated algorithms, artificial intelligence algorithms for automated detection. With regards to schistosomiasis, we have developed a device called the schistoscope, and there are 5 versions of this device, and these iterations have been reflected in a conference paper. For sample preparation, we will use urine filtration. We are also in the process of developing dedicated algorithms for automated detection. With regards to hookworm, we have explored an automated fecal smear device. And as you can see in the image on the bottom right, it's a very complex device. So we have decided to continue with the standard Cato-CAT slides. As you can see, there is a multidisciplinary team that developed the schistoscope. And this device also won the NTD Innovation Prize in 2021 in collaboration with the startup ADEX. So in this image, you can see the device, you can see the students that have been developing these devices. You also see that it's packaged here, ready to be sent to be tested and validated in the field. Of course, there are other tests to detect schistosomiasis, and two prominent examples are the UCP-LF-CAA test as well as the POC-CCA test. And if you would like more information on these tests, there is also another video on our website. In terms of technology development, we are also exploring two case studies of strip readers. We've developed a prototype with a bachelor project for the POC-CCA reader. And we now also have a junior researcher who is exploring the development of a reader for the UCP-LF-CAA test. In terms of education and training methods, which was an interesting outcome from work package 2, we have also developed the EduScope. And for this, 2 master student groups have developed the hardware as well as the user interface. The EduScope was nominated for an award, the New Talent category, and it also won the UK Braun Prize Brightest Design Talent in 2021. And this Braun Prize will be used to locally produce devices in Kenya and validate its use in an educational setting, such as a high school. Work package 2 deals with an ecosystem analysis. There are many levels, organizational levels in a country. On a micro level, there are small systems such as families. On a meso level, medium systems such as organizations. And on a macro level, you have larger systems such as the legal systems in place or on a national level. And we chose to start with Nigeria as a case study because there are 220 million people, so it's a really big context. And schistosomiasis is highly prevalent. So what we do in work package 2 is a stakeholder analysis at all of these different levels of the ecosystem. And we use co-creation tools such as a social network analysis. This allows us to identify relevant stakeholders. And with those relevant stakeholders, we can do further research using co-creation tools such as Q methodology. And the idea here is that the outcomes of this research would contribute towards a target product profile for these innovative optical devices. As you can see, it's a multi-layer project with many people that includes field assistants and researchers. And it really is a paradigm shift from the technology push. So instead of devices being developed in Europe and being expected to work in the global South, we take a very different approach with co-creation, where we want to sensitize, involve, and facilitate a pathway to adopt the new technology. Here you see an impression of the co-creation and the research that is being done in the field by many different partners and research assistants. So work package 3 deals with laboratory and field validation of the devices that are developed in work package 1. And a large part of validation involves biobanking, so that is sample collection. And we are also planning an extensive validation of the work package 1 devices in field settings. So we also test prototypes in this work package in field settings in Gabon, as well as laboratory settings in Gabon and the Netherlands. In terms of the schistoscope, as an example, version 3.0 was tested in Gabon, and the feedback from that was sent to the team in Delft, where they then developed version 4.0. Version 4.0 was then tested in Nigeria, and while version 5.0 was being developed, and version 5.0 was also recently tested in Nigeria. Work package 3 also contributes towards sample preparation in terms of optimizing techniques for the devices that might be in staining slides, as well as preparing samples, so spiking eggs, and this is an important step in training the automated detection algorithms. So the scope of this work package is not only limited to the validation of the devices, but also about adapting reference tests for general diagnostics, for example, using PCR in a composite reference standard. So this is an impression of the field validation of the Schistoscope 5.0 that was recently conducted in the summer 2021 in Abuja, Nigeria. We tested the performance of the Schistoscope compared to conventional microscopy. And of course, a multidisciplinary team was involved in this validation study. Continuing with work package 4, which deals with implementation and scalability. In terms of new education and training methods, we realized that there is a need for a transition to digital optical devices. And for this, we would like to use the Eduscope device to perform a next level implementation and upscale analysis. And we're also looking at stakeholder analysis for end user requirements in different settings. So much of the work for the Schistoscope has been dedicated to public health settings, as you saw in work package 2. But we also have the unique opportunity to look at a clinical trial setting, and that is in collaboration with the EDCTP STOP project. And so with this collaboration, we will be exploring stakeholder perspectives for optical diagnostic devices in research clinical trial settings. We also do a lot of networking and stakeholder analysis meetings to introduce devices in the policy agenda for poverty-related parasitic diseases in Nigeria. And we currently have a PhD and postdoc in Ibadan, Nigeria in November 2021. So here I would like to highlight 2 case studies, specifically the Eduscope and the strip readers. And this is towards the development of a dedicated UCP LFCAA reader and or an RDT reader. And that could be for an RDT, that's a rapid diagnostic test for other diseases as well, such as malaria. So the image on the bottom right, that is a very early prototype of an RDT reader that we are working towards developing. I would now like to highlight our publications and other outputs. We have so far have 5 journal articles. We have published 5 conference papers, many master theses, a bachelor in project, as well as a PhD dissertation. And I highlight here a news roundup where one of our publications was highlighted in March of 2021. And this is the newsletter from the Neglected Tropical Diseases Support Center. We have hosted 2 symposiums, and we have a very active website, and we are also active on LinkedIn. If you're interested in any of the symposiums, you can also find this information on our website. And to end, I would like to acknowledge all of the researchers and all of the partners that have been involved in this research. Thank you.